Hi everyone, tonight I'm sitting down with John Oliver to talk about insulin receptor regulation. Let's get started.
Science is constantly producing new studies, as you would know if you've ever watched TV.
A new study shows how sugar might fuel the growth of cancer.
A new study shows late-night snacking could damage the part of your brain that creates and stores memories.
A new study finds pizza is the most addictive food in America.
A new study suggests hugging your dog is bad for your dog.
Yeah, but insulin receptor regulation is a bit more important than that. Look here. The reduced biological response to insulin by tissues results from an impairment in the cascade of phosphorylation events within cells that can regulate the activity of enzymes compromising the insulin signaling pathway. This begins with either the activation of the insulin receptor or substrate kinase activity of the receptor, and can lead to type 2 diabetes and the risk of cardiovascular diseases.
We love food in America, as you would know if you've ever turned on a television set.
IHOP all-you-can-eat pancakes are back.
Introducing all-you-can-eat wings only at Golden Corral.
Riblets. Applebee's has riblets. All that you can eat.
Yes, John, it sure does make it hard for us to keep our insulin sensitivity regulated if we aren't keeping up with our proper dietary habits and physical activity. Back to what I was previously saying. Activation of the insulin receptor can be impaired by post-translational modifications of the protein involving serine phosphorylation or by binding to inhibiting proteins. This can impact the conformational changes in phosphorylation events required for full signaling activity. During insulin resistance, the substrate tyrosine kinase activity of the insulin receptor causes diminished ability of the insulin to initiate these phosphorylation cascades that regulate the insulin second messenger. Evidence suggests that acute and chronic regulation of insulin receptors function contributes to alterations in insulin sensitivity and glucose homeostasis. There are also mechanisms available and used to regulate the IR function. These processes involve either modification of IR content in the cells at the level of transcription or protein degradation, or by modification of enzymatic activity of the individual IR. Transcription factors that regulate the IR content in vivo are still not well understood. However, Puig and Tejon have proved that the transcription factor FOXO1 mediates the downregulation of IR transcription in a response to the stimulation of insulin in the IR signaling. When insulin is absent, such as during times of fasting, FOXO1 binds to the IR promoter and can stimulate the IR gene transcription. This stimulation can result in phosphorylation of FOXO1, which disrupts the IR promoter interaction. What? Are you bored? Think you can do better?
We have you covered.
Do you love science in all its complexity, but wish it could be a little less complex and a lot less scientific? Introducing Todd Talks, where the format of TED Talks meets the intellectual rigor of morning news shows.
Chocolate. It will kill you.
In my research, I found out red wine makes babies 20% more sociable.
On that note, I think I'll hand it over to Sam.